This year we decided as a group to have our largest focus on Matlab as a tool. And I think a lot of leadership in the state has been shown by Sumeet and Pradeep with this. This is also a tool that Urska and Marian in earlier days had made strong investments. As a core, we're also making some software and, well, at least software investments in making some of the underlying tools that you're seeing available on some large compute resources to the full LBRN community. So with that, the first 45 minutes, Pradeep and Sumeet will be presenting, and then I'll give a little bit at the end to talk about some aspects of what's next. Looking forward.
Right. I'm Sumeet Dua. That's Pradeep Chowriappa. We're going to be talking about Matlab. In fact, primarily Pradeep is going to be talking about Matlab. The objective here is to actually give you a feel of what Matlab is all about, how it operates. It has toolboxes, so we are going to be talking about the Bioinformatics toolbox, which also uses the statistics toolbox in Matlab. The objective is not to give you hands-on understanding of how to program in Matlab. And there are several resources that you can tap to understand that. But the objective here is to actually tell you what these toolbox can accomplish. They can do sequence analysis. There's some very nice microarray gene expression analysis tools, visualization tools that you can leverage. So you're going to be hearing about those as well. Later on, in the last 20 minutes or so, we're going to be talking about high-performance computing, how Matlab can operate on a parallel environment. Matlab can get very slow at times when it comes down to high-dimensional data. So it's really important to parallelize the programs, and we have done some efforts in that area. There's integration with GPUs, or GPU, general purpose graphical processing units. We're going to be talking about those, and then we'll also tell you about the parallel computing toolbox in Matlab, which is kind of their solution to parallelization of Matlab. We have also used a resource, a middleware resource called Jacket, which is not from MathWorks, which is the parent company of Matlab, but it allows you to use Matlab on a GPU environment. So you'll hear some about that as well. Feel free to ask questions as we move along. A lot of this content is available on MathWorks website and so on, but then we'll also have some examples of how we have been able to use this in research.
I think he said everything that I had. Well, okay, let me go in depth. Apart from, the objective of this talk is actually to create an awareness of using Matlab in research. Now, there are different flavors of programming that can be exploited for bioinformatics, different flavors of tools out there that can be exploited for our research. But why did we choose Matlab, and why is it of importance, and why it can be exploited in our research areas? So that is the talk, what we're focusing on today. So what is Matlab? I'm sure everyone knows what Matlab is. Matlab is actually a proprietary software that is created from MathWorks. It is a high-level interface, a technical programming language that provides numeric operations. It enables algorithm development, and it provides neat visualization and data analysis. Moreover, it comes in all the three flavors. So it could be for the Windows or Mac or Linux. So it can be installed and it can be run on any of your computers. So that's just the overview of Matlab. And this is a screenshot of how Matlab looks like. So it works on the principle where it gives the user to neatly interact with his so-called variables. It has a workspace that can be exploited by the programmer to find out what he's using. Okay, it does enable you to do both coding as well as command line interface. So it has something called the command window where you can do a quick execution of a command apart from writing your usual code. Okay. So before we understanding what Matlab is, it's nothing else but Matlab was initially created as a wrapper around Fortran, and Fortran. It had a flavor of a language, but later on evolved into an environment. So there are two perspectives towards Matlab. One is the programming, and the other one is the environment that it actually gives the user. So there are pros and cons of Matlab when you compare it with Python and NumPy, but when you go towards the programming perspective, Matlab is not that fancy. It doesn't. If it is a hardcore programmer from a computer science background, he would definitely prefer more of Python, as well as the NumPy options. But what MATLAB gives you that's way beyond that of Python and NumPy is it gives you an environment which enables a novice user, say for example, a user from the engineering background or a user from the biological background who wants to investigate how to use a programming environment. MATLAB gives you the feel and the ease in which you can try out what you want. So it has, with MATLAB, that relies over the computational resource. It could be a single CPU or it could be a parallel environment. It does give you flavors of toolboxes. Now that is where MATLAB and MathWorks has invested immense amount of time. They have neat documentation, which it's very easy. If you were to go try the Python, I'm not trying to discourage the use of Python and NumPy, but it is, if you use it, you tend to realize how difficult it is to actually know what you're doing, because you don't have effective documentation, and that's where if a biologist had to enter who has no idea about what are the functions that Python and NumPy has, MATLAB provides you the opportunity to, with its immense documentation, to find out where what is located. So they have structured different toolboxes, and primarily we have the statistical that gives all your statistical functions, over which you have other toolboxes that leverage from the statistical toolbox. So you have the bioinformatics, you have flavors of signal processing, you have image processing, then you have even Simulink. This is all integrated into one tool that is tied down with MATLAB. Now, if you were to compare MATLAB with Mathematica, on the other hand, Mathematica is, and I mean, you just can't compare the two in the sense, this is more of a quick prototyping environment, whereas Mathematica does give you the opportunity to understand the crux behind an algorithm, say for example. So there are pros and cons of both. But if you were to try MATLAB, it does give you the additional documentation where a novice user can quickly traverse into the toolbox and find out its different functions and the associated papers and the associated usage of how to actually implement that so-called function into your code. So the focus of this talk was actually to create an awareness of the bioinformatics toolbox. So, as I mentioned previously, it has a lot of its functions that rest on the statistical toolbox. So, the toolbox effectively enables the prototyping where you could create your own GUI. You could just try out for a simple project. You can pull in different functions and create an effective GUI where you can just submit a sequence maybe and then see the potential outcomes. So it does give you the share. These applications, on the other hand, can also be shared across different platforms. So you can, once you create this prototype, you could actually share it and visualize it effectively. Now, its strengths, as per the bioinformatics toolbox, its strengths lie in the functions that can be used and exploited for sequence analysis, microarray data analysis, proteomics, for example. It gives you very useful methods and techniques that can be used for analyzing protein structure and sequence. Apart from that, it does have simple mining approaches like the simple k-means and the hierarchical clustering that can be exploited, as well as it does give you this, the hidden Markov model profiles where you can actually compare and see different sequences and how they align with each other. Okay. So MATLAB, the data formats and the databases. So MATLAB has invested a lot into providing the user the access with a simple command to the different databases out there. So for example, if you have web-based databases, it gives you access to the GenBank, the GenPep, the EMBL, the GEO database, all with a single command, as well as the PDB. So what it does is all that you have to do is say read from the PDB, give your protein ID, it will extract the file from the database, and it will give it to you in the format that it is saved in. So it also gives you access to the multiple hidden Markov model profiles that can be exploited for your analysis from the PFAM database. It gives you access to the gene ontology, so you could find the functional information that is required for a sequence. Maybe you discover something new and you need to know the ontology associated with that. You could very well connect to the gene ontology database. And it supports the reading and writing. So you could read a BLAST file, you can read and you can save it in the format that is required as well. So it does the blast searches as well. So all with a single command. I will show you an example later on. So when I was preparing this slide, I just wanted to give you an overview. So apart from the different functions that the Bioinformatics toolbox has, it also has inbuilt tools available. For example, this is just the example of the sequence tool that is available provided by Matlab. It does allow you to directly connect to the NCBI database, where you can download your given genome sequence data with the NCBI ID, the accession number, and it will load it, and it will give you all the different associated functions that can be associated with that, all in a simple GUI. But it also gives you the leverage of using its commands that this tool is based on. So it gives you the overall feel, plus it gives you access to the functions that can be performed on the sequence. It helps you, one of the unique features of this tool is actually it helps you determine the consensus sequence if you have multiple sequences. So that sometimes if you have to compare them, it gives you the consensus sequence. So say for example, we have the end density for a given genome sequence. With a simple command of just giving the end density with the accession number, it could give you the distribution of the nucleotides, okay, the density. Then it also gives you the base count. These are just simple as possible. It's just give a command and it would generate to you the so-called distributions. So it gives you a neat idea of how the base counts are distributed given the given genome sequence. It gives you the dimer counts as well. It gives you the bar in a bar chart. This is just the simplicity in which it enables you to visualize data. This is another representation of the codon counts. You can give the sequence and it would represent the codons, the different dimers and the codons that are there for the given sequence. And its frequency of occurrence as well in the matrix form. So you can visualize as well as you can extract data. So you can do those operations. So this is another, it gives you sequence alignments. It gives you an option of using both the Needleman and Wunsch as well as the Smith and Waterman's algorithm. So you can do your comparison of sequences, alignment of sequences in both flavors. And it gives you the neat outputs where you could actually see where they align and misalign as well. So when it comes to, this is where we invest a lot of Matlab. We use a lot of Matlab for microarray analysis, where it gives you the flavor of pre-processing, it gives you the ability to clean your data, your microarrays. It then gives you most of the prominently used normalization strategies that can be used for gene expression data. So these are reliable techniques, they've been coded, you need not recreate that entire algorithm or the philosophy behind it. All you have to do is invoke and it will do it for you, okay? Then it also provides useful visualization of these data. You have the heat maps, you have the log-log plots, as well as you have the IR plots. I just have some screenshots of that. So this is just the heat map view of in a microarray image form. So you have a spatial location of all your expressions for a given microarray distributed in values. So you could actually visualize that, the entire microarray experiment in a single view. Then it gives you the microarray box plots. You can actually see the distributions of your expression values. So this was just a screenshot. These are all from the help. This is all the documentation provided in Matlab that can be used. It also gives you to do a principal component analysis to identify outliers, for example. You can very well do an n-dimensional view of your gene expression data, and you can identify outliers. You can remove them, you can keep them, you can identify clusters of interest that can be exploited for your analysis. So PCA2 can be done very easily with Matlab. The entire philosophy of Matlab is to work with vectors, and this becomes very simple unless you try to do it otherwise. So Matlab works on that philosophy. You can do cluster analysis. They have options to do hierarchical clustering, where you can understand expression profiles and cluster them based on their profiles. You can visualize the same hierarchical in that form, as well as you can do your dendrogram, where you get clustered them both in sample space as well as your gene expression, gene space. You can do both in the clustergram. This is another clustergram, is another tool that you can simply invoke with your data, and it will generate the useful visualization. It has also functions associated with phylogenetic analysis. You have to represent it, say if you have a bunch of genome sequences, you can see the similarity, the regions that they match. The similarity matrices that it works on is the Juice Cantor, the p-dist. The alignment scores can be exploited to generate your phylogenetic tree, as well as user-defined distances. Then you have visualization. It has an inbuilt functions as well that can be exploited to visualize 3D structure. You have the mall viewer. This tool enables you to visualize as well as structurally align two structures. So the small viewer has other associated functions as well, which can be exploited to have an in-depth view of your entire protein. So now we move over from the entire. Those are the flavors. I tried to encapsulate this huge thing in a set of screenshots. But if there is interest, you're more than welcome to refer the documentation that is provided by Matlab. They have neat demos, and it would give you an in-depth analysis of that. They're moving over to the next-gen sequencing also, though not there, because the entire philosophy of Matlab is based on matrices, and it would eventually run down to the bottleneck of storing and that data onto your local computer. So, there are functions available that can be used on smaller data sets, but typically when it's the next gen, you need to. They have examples and demos of exploiting Matlab for metagenome analysis as well. It does have this phylogenetic view of your metagenome data. It has functions associated with that as well. So now we move over to the parallel concept and how Matlab is moving away from their so-called parallel computing towards more GPU, and how is it going to change everything. This section was actually to give you a small overview of how Matlab can be exploited for parallel computing. So I'm just going to breeze through the serial. I'm sure most of you all are aware of the difference between serial computing as well as parallel computing. So if you have a job, you could very well split the job into multiple instructions, and each instruction is executed sequentially, one after the other. That is an overview of just plain serial computing. But when we move over to the parallel computing, you want to make sure that you want to split these processes and these instructions on in a distributed environment where you have multiple CPUs, not just one CPU to execute instruction one at a time. So the biggest challenge over here it's not easy for a computer scientist as well, but I believe it's even more challenging for a biologist to even visualize how I could split a job to suit a parallel execution. So this is the bottleneck, and this is where many fear the distributed environment concept. So the challenge here is to actually break down your entire sequential into independent, let's not go into that, but into parallel execution steps that can be run in parallel, such that you save time and exploit resources. So in the simplest sense, parallel computing is like running multiple simulations of different computers. So you could have different inputs for the same set of commands and run it on different CPUs. Or you could split one big program and run it independently on different CPUs. So these are just a simple view of an overbistic view. My idea here was not to discourage, but to exploit what we already have and use what we are going to foresee in the future. So this is to dispel the so-called fears that many of us have. And we need not go through intensive parallelization, though required. We need not go into intensive parallelization. Before we're thinking about splitting a large task into subgroups, we do have to take into consideration, this is an important topic which would enable the GPU computing, is to understand what is the memory and how it can be exploited. Okay? So typically, what is ideal is we have this uniform memory access where you have this huge memory pool and you have different processors that can exploit this memory. Now, this is an ideal scenario where you have this uniform pool. But now, when we move more over into GPUs and parallel environments, this is more where you have a set of CPUs accessing multiple memories. So if you have something updating a memory operation here, this has to be, you need to tell every other CPU that's involved in the entire network. Okay? So if you're distributing, you need to you need to keep in mind the different architecture that's involved. Now this is where the bottleneck comes in. We have technology that's immensely growing in the biology field. Now we have technology also moving very fast and rapidly into the computational field. We had regular sequencing. Now it's going towards the next-gen sequencing, which is going to be an implosive. I mean, it's going to have an explosion of data. And how do we exploit it with the technology that's equally growing in the computational field? We had the distributed environment, and now we're moving into the GPUs. So, and how does Matlab facilitate all of this? So, parallel computing with Matlab is simple. You have Matlab, you have its different toolboxes, you have the important component of Simulink, which helps you do simulations, as well as you have your workspaces. Now, how do we move into a pool of computers that host Matlab, and how can we exploit this distributed environment to execute a single job? Now, that is the challenge. There are different aspects to job parallelization. The most common, of course, will be the pool worker or a bag of tasks. So the idea here is to understand your entire workflow and divide it into independent tasks and execute them one at a time. Now that is the simplest way of doing multitasking. Maybe you have different inputs, try out independent tasks and distribute it effectively such that you save time. Okay? So, job parallelization is more of, what I wanted to say over here is sequential parallel. So, where you have just to take your sequential code and effectively just mimic that on different CPUs. So, in that way, typically that's what we face with how do we effectively do this. And this is where maybe we can aid a biologist from doing parallel computing. But all that we need to know in this is what is your inputs and what are your desired outputs. You do save your resources, exploit it to the maximum, and in the end, you get effective time utilization. That is our objective. Okay? The biggest bottleneck in Matlab is this iterative for loops. I guess a for loop is running multiple times for a fixed number of iterations. And when you have an iteration within an iteration, this is the bottleneck of actually this is a bottleneck of actually doing effective parallelism. So this is where you need to really think about your code and see how you can exploit it to do effective parallelism. And this becomes even more important when you're handling GPUs or any parallel environment. So what is GPGPU? GPGPU is nothing else but you have different multiprocessors on a single card. Ideally, it was created for putting this onto a computer or your laptop to get effective visualization, faster videos, better gameplay, and it was created by NVIDIA. Now, what they realized later was this entire architecture that they had come out could be exploited for your tasks, where you could actually use it to do faster computing. It was like putting a supercomputer onto your local desktop. So CUDA and NVIDIA have this architecture. They have all these different multiprocessors on a single chip, and they came out with different flavors of this architecture. You have the Tesla, You have the Quattro and you have the GeForce. Right now, Matlab supports the NVIDIA CUDA architecture and the libraries, but it works on the Tesla 10. We have the Tesla 20s, 20s, and the 20 series of GPUs. Now, Matlab had something called as the parallel computing toolbox. Now, this toolbox is moving towards the GPUs, where you can just invoke all the parallel commands with a simple GPU command before it. So they're moving right from a distributed environment directly onto the GPUs, so that can be exploited. So this is an architectural comparison. If you have a CPU, you have multiple cores, and each core can be exploited in a distributed environment. So if you have a 4-core computer, you can very well do distributed computing on your existing computer. Yes?
Can I just add, Pradeep? I don't know if everybody knows the GPU is on the graphics card, the
Yes.
graphics processing unit. So I just wanted to add that because
No,
I know there's
I-
life scientists that
Oh, okay.
didn't know what is the difference between a CPU and a GPU, I bet. So CPU is your, core chip that you have on the motherboard to process things, and the GPU is the chip that is on the graphics processing unit or your graphics card that also does displays. So, and most of the time, if you wanted to do this, you probably have to buy a graphics card in addition to what you currently have on the machine. It's not coming with a basic desktop computer.
You would need that. You would need the Tesla card. And you could have your existing GPU visualization cards that can be exploited as well, but you need the Tesla cards, which are actually independent sockets that could be put onto your computer.
And well, more practically, actually, you need a GPU optimized system. It's not simply the matter of buying a Tesla car and putting that into a standard machine. The GPU machines that we have, they're not from any standard vendor like Dell or IBM. It's a third-party vendor that assembles these machines. They optimize them. They ship those with some software that helps with the optimization process. Just to give you an idea, you can actually get like a theoretical capacity for about 1 teraflops for less than $5,000 with that third-party vendor. But then it's theoretical, and I'll put that in bold and underline.
A small variation on what's being said. There are two sorts of genres of computing, I think, in general, people here are doing at the sort of PC scale, something under your desk, or at larger scales. And at the PC scale, the last 5-7 years, adding these GPUs has become very, very popular. But it's not been until the very most recent time when the GPUs are actually starting to make it into larger clusters.
Right.
And so right now, as of this moment, in the Lonnie system going throughout the state, I don't believe there are any large compute clusters that have GPUs. But just in the next few months, some of the first large ones are coming in, and I think as an example, the Blue Water is one of the largest systems in the country. I haven't seen it now that IBM backed out of the bid and it's moving to Cray, but originally it was to be like 30% of the machine was to be GPUs, and 70% the sort of more conventional CPUs, just because of some of the performance gains that people have already been realizing on the smaller scale systems.
One more distraction. So we put in our first GPU cluster at UGA. It's only like 10 nodes, and each one I think has 6 GPUs on it or something of that nature. And it's wickedly fast. It's really nice. But the bottleneck is not getting on here. It's being able to program in CUDA. So NVIDIA, we were able to apply and we were awarded to become a CUDA training center or something like this. So I highly encourage people here to do that because not all of a lot of code, blast and things we might use, a lot of already been ported. But a lot of applications haven't, and so until we have a generation of people who know how to write a CUDA.
Again, as Ma'am said, CUDA is intensive for people who love programming. You need to fall in love with it. But you also need to wait. You also need to wait to. Once you get to see your outputs, they are static. But then it comes to quick prototyping, and that's where a lot of us, especially students, who have to exploit a huge resource, we find Matlab and Jacket fitting that gap very easily.
From proof of principle
For a
standpoint,
proof of principle.
like, as a stepping stone for a student trying to move up to an HPC environment from a desktop environment, I think this has really done well for us in that respect. About 2 years back, I decided to replace all of our desktops in my lab with GPUs, and there was no Microsoft Office on it. I said, If you have to program anything, you have to program it on a GPU, period. And it really helped us. I think each one of our members in the lab actually now can do GPU computing for very basic programming, even class assignments. That really helped us.
I mean, and now Matlab is catching up because they've come out. You need not go through Jacket anymore. You could just invoke. I will explain that. I mean, Dr. Urska actually pointed that out very easily. I kind of overlooked that. When it comes to GPU programming, I mean, this slide was supposed to capture that. You have cores that you can do your distributed environment, but you can add the GPU to it, where you can exploit the different cores that the GPU supports. Now remember, I mean, not everything is 100%, and it's just not a plug and play that you can do. A lot of the communication occurs between the CPU and the GPU. So your memory, your RAM on your local computer, where the CPU should be high, because that's where, and this is where it gets hot, in the sense you need to do a lot of talking between these two. So you need to transfer what is there on your RAM or through your CPU back to the GPU to do its computation and send it back. So you face this challenge with the GPUs. And
I think that, and so here's a very interesting point that Pradeep made, that that's one of those programming things that you have to be really careful that you're not actually allowing the program to communicate back and forth a whole lot between the GPU and the CPU. If you do that, you actually lose the benefit of getting onto a parallel environment.
Yeah, and
so that's a programming skill that you will master over time. Where you'll try to minimize that communication, which is kind of true for any HPC environment. You shouldn't be spending too much time on IOs. You should be kind of processing more on one single core or a processor.
And that's why the first half of my talk was actually not to get it too complicated, but keep it as simple as possible. All that you have to worry about is let's distribute it in a simple way, rather than trying to worry too much about it, because a biologist would definitely find it challenging. And that's where, I mean, yeah, okay.
Yeah, 5 minutes.
Sorry. So this was a nice comparative view as to the time that you could actually save. You have with the Intel core, and this is the GeForce, this is the GPUs when you compare that.
So the longer the bar, the better.
Yeah, the longer the bar, the better. This is the whole chromosome.
Number of samples processed per hour.
Yeah, number of more.
The more you do.
So, okay, I was coming back to something that this is for using data analysis in CUDA, and this is where we had the Matlab standing out with its parallel computing toolbox. I mean, these were some of the prominent tools that can be exploited for data analysis. You have the parse stream. These are prominent tools that you can really check on if you are interested. Some of our students actually did that as part of our workshop. You have the jacket, which was third, that can be exploited, and that's where we wanted to. This is yet to catch up. Some of the functionalities have to be really built on when it came to. This was actually 3 months ago, or way back in October, that Matlab actually announced that they're moving on to the GPUs and trying to change the entire parallel computing toolbox towards that. You have Wolfram's Mathematica there, but these are some of the over-the-counter tools that can be exploited for GPUs. You have the libSVM, where you can do supervised learning with the GPUs. Then you have R-based tools, where you have the R, EUSUM, and the CUSUM with the CUDA SVM. Then you have these were the prominent ones. You have the GPU miner, where the entire architecture and the entire philosophy of doing data mining with GPU enabled. This is the GPU miner. We have the CMatch, and we have the R-based, which is yet in its preliminary stages, and they are going to evolve. And they are going to. The thing is, they are open source. This one, and you can exploit that. Yes. So you have GPU auxiliary. I have some examples here. Do you want me to go through examples?
I don't think we have time. I think if you can, in the last 2 or 3 minutes, sort of wrap up, just trying to give people really a taste of some of the spaces you've been covering.
No, I just wanted to just give them a flavor. Matlab came up with the GPU arrays, where you can actually port. The idea, as Dr. Urska said, moving from CPU to the GPU. That's their philosophy. And they invoked something called as the GPU array, which is nothing else but transferring your CPU data onto the GPU. And then afterwards you have the gather operation. Whatever's processed on your GPU is back again to your CPU. Now these are the only 2 constraints that you need to worry about when you're transferring data from CPU, GPU to CPU. And these are the important functions that you really need to worry about. Okay, this is some of the library functions where you can directly invoke. You need not transfer your data to see what's there from your GPU. They have a function where you can just say, show me what's there, or you can directly create an array on your GPU. They have something with simple commands like that. Let me come to this. So this is where we are right now. These are the set of functions that it actually can support. This is the functions that GPU array can support with your GPU. They have yet to catch up with what are the functions that are there on the parallel toolbox. So this is still in the initial stages. When you have to exploit your so-called Matlab for GPU operations, you can just do it with this set of functions. Now, exploiting a library, or say Bioinformatics toolbox for GPU is still exploratory. We've yet to try that out effectively. Now, this is the overview of the jacket, the tool that we use in our lab. If you go to Matlab and the parallel computing, you need to go through all of these operations.
So let me put this in perspective. The way Matlab operates is that you have a core set of libraries which are part of the Matlab software. So if you just were to buy Matlab, you are not using any toolboxes, you will have some libraries that you can use in Matlab. The functions that was listed on the previous slide are part of that library.
Yes.
They're not part of the toolbox. Then if you were to buy a toolbox like Bioinformatics toolbox, some toolboxes have dependencies. So if you buy Bioinformatics, you need to have statistics toolbox installed prior to getting Bioinformatics installed. None of these functions are in those toolboxes. There are some people have developed, they have posted those on what's called the Web Central, the Matlab share site, but none of those are being sold by MathWorks, the parent company. And that's where Jacket comes in, where Jacket completely ignores whether you're coming from a toolbox or whether you're coming from basic MATLAB, because it's a layer beyond MATLAB. It's like a middleware. Okay, so that's the reason why Jacket becomes useful in addition to what MathWorks is providing you, the parallel computing toolbox.
It actually shadows this entire route that you need to take care of if you're doing with parallel computing. MATLAB Jacket just overlooks that. So that made it easier. That made students from non-computer science background to actually exploit the GPU in a very simple fashion. So that's my contribution, our talk. You can look at the references. Most of it is out there. You just visit MathWorks and you can get it.
Wonderful. Oh, you can call this guy. He's very well trained and he sleeps and breathes MATLAB.
Well, and this is a day's worth of material to try to whiz through in 40 minutes, but Pradeep has been very active and effective in making use of Pradeep, let's take a couple questions from folks.
Yes.
Well, I was curious how the development for under the new framework for the GPU and MATLAB compared to like OpenCL. Because for instance, like at OpenCL, I can write a kernel for a variety of different targets and reuse that same kernel. Could I do the same thing with MATLAB?
MATLAB allows you to do everything what you do in writing. Writing your code, you could write it the way you write it in regular fashion. All that it enables you is to exploit benchmarks functions. You need not worry about implementing the Needleman's alignment algorithm all over again. It's just like, say for example, if you're using Python and you are confident. Are you confident with borrowing a function? And that's where it's easy for us. I mean, we really need not worry about it.
And you can also create your own library. So not only you're relying on what's being supplied to you, you could also create your own functions, and then that wouldn't become the part of the toolbox, but that would be a part of your working library that you can use. And then a lot of students will do that. You know, they will try to create functions. Like in any modular programming environment, you should be allowed to do that. So it helps you with that. The ease, like Pradeep said, the ease here is that you are trying to borrow functions which have been tested, evaluated, scaled for certain functionality.
Other questions? Well, let me ask a question as we transition. So, let's say that you are approached by someone in LBRN and they're developing a new proposal to LBRN, or better yet, an R15 or something like this. They know that they're going to need to deal with some large data sets, but they don't have a lot of in-house sort of computational expertise. They know that they're going to allocate money for a graduate student on their team or an undergraduate student. and they're really excited about what you've been describing with Matlab as a way to get going, to begin engaging with parallelism without needing to have 3 postdocs and blah, blah, blah. It's hard to say in general, but if you're trying to say the first few words of advice in terms of how they're going to train that student, what's a good kind of warm-up problem to begin analyzing and visualizing some chromosomes in some way, they're wanting to have some sort of warm-up to begin touching the GPU aspects. It's again really hard in general question, but you have someone and they're trying to flesh out the proposal and trying to go to you for advice on how to think about the road ahead. Any first impressions?
I mean, it's the same philosophy that I spoke to you about, which I try to imbibe with my experience for 2 years with the LBRN, is to reduce our scope, to make our case, to show our hypothesis, and then increase our scope. That's the philosophy we did. And many of our PIs actually come with huge amounts of data. They talk about teraflops of data. Now, when they approach me with huge amounts of data and they ask me to come out with an idea, my first instinct is, let's try to understand the problem and reduce the scope. Parallelism can be limited. The minute you show that it is doable, and then that's when they get confidence, and then we can move on with distributing it. That's what I follow.
So, if the question was more about Matlab versus high performance computing or Jack environment, there are a lot of good resources available from MathWorks. Toy problems, examples, data sets that you can download and execute for various different toolboxes. We have also enjoyed some good videos that they offer as part of the training sessions. There's a lot of free training sessions that you can actually subscribe to, and these are webinars that happens every 3 weeks or 4 weeks and so on. So those are good ways to get warm up in how to use Matlab, how to use toolboxes, understanding the functionality of toolboxes and their functions.
And have either of you found a favorite Hello World you would commend to people, of chromosome 15 of the human reference genome,
Yes, sir, I totally understand. For you to get a perfect flavor of Matlab, use the demos. They have inbuilt demos. They give you a step-by-step walkthrough of how to use the basic...
And to this audience, do you have a favorite one of the demos, or a favorite one of the warm-ups dealing with chromosomal data, or second-gen data, or other
Next-gen, they do have a demo on next-gen. Especially, they have 3 demos on next-gen sequencing where you can exploit their simple basic functionality of reading, let alone reading those huge amounts of files. They give you an example. They have inbuilt benchmark data set that can be exploited on any computer. Exploit their functionality. They have a very good demo for metagenome analysis where you have multiple next gens together, all together in one. And it does the neat phylogenetic analysis. They do the alignments, assemblies. They have all inbuilt functions. Now again, they can't compare with what is out there for next gen. You cannot compare the performance of Abyss with Matlab's implementation. The philosophy is totally different. We can't compare it with what Bowtie has to offer. But it does have functionality to understand and visualize, which is important for a computer science and an engineer.
And that brings us back to the original point that I was making. There's very important proof of principle concept. Matlab is really good for you to establish scope, to establish your algorithm, establish the logic of operation. But when it comes down to scalability, then you have problems. Primarily because the way Matlab is designed is matrix-based.
It's matrix-based.
And a lot of these matrices tend to be sparse, that waste a lot of computational time executing. So if you have like 200,000 dimensions to deal with, you're talking about 200,000 columns to an array, and a lot of which may not be may be totally empty, and you'll be spending a lot of time computing or differencing matrices.
It's good for prototyping. Our offers the same flavors of Matlab. I mean, same almost, but its philosophy is different. I mean, if you're intensively into coding and you want to exploit the statistical, you could very well exploit R. Their libraries are just as good.
And I'll also add that you can export Matlab code into C, although I would strongly recommend against it. You come to a point where you say, I would have been much better off writing this from scratch, rather than exporting it. But it is possible to do.
Well, thank you very much, Pradeep and Sumeet.